It's a great pleasure to introduce the next speaker and the last speaker for this session, Dr. Claire Pichon, who is an investigator at the National Institutes of Child Health and Human Development at the NIH. Claire did her PhD at Columbia University, and then she got recruited at Genentech at the neuroscience department there, where she worked for several years with Joel Leacock and others. And she directly contributed to the identification of DLK inhibitors that made it to clinical trials in the context of neurodegenerative diseases. She's been at the NIH now for a number of years, 7 years, I think. And she is interested in understanding several pathways associated with degeneration, such as axonal injury, mitochondrial dysfunction. And I'm really delighted that she's here. Looking forward to her talk. Welcome.
Thanks so much for the introduction, Evangelos. And let's see. So to advance the slides, press this. Okay. The forward. Oh, down. Okay, great. So I'm going to talk a little bit about something, I guess, potentially more downstream than everything we've been hearing about today so far. Whoopsies. And what my lab focuses on is really understanding pathways that regulate axon degeneration and regeneration. And I think this is highly relevant in the context of lots of neurodegenerative conditions, including ALS. Just an overview of the types of techniques we use in the lab. We do a lot of mouse modeling, including kind of the newer type of mouse modeling, which is to express a mutation that would be found in a patient and put that in the mouse version of the gene, because very often the genes linked to neurodegenerative disease are highly conserved. And then we, as Evangelos mentioned, we leverage more acute models of neuronal injury, including something we heard about from Daryl, so traumatic brain injury, but also traumatic nerve injury. And then recently we've forayed into kind of using single cell transcriptomics to try to better understand cell type specificity and why are some neurons more affected than others in disease. And I apologize because I kind of advertised this talk would focus on that, but it's not. But there's, I think, I'm going to get to it at the end. And then finally, we're also using iPSC-derived human neurons, which you heard about from various talks, especially Evangelos introduced this very nicely, and also Daryl. So this is really great because I don't have to re-explain everything. Okay, so kind of in the most basic fundamental sense, the question that I originally started working on when at Genentech during my postdoc, the equivalent of my postdoc years was, okay, we have all these different neurodegenerative diseases, and yes, in some very small number of cases, we know a gene that causes the disease, but most of the cases we don't know, we don't understand, and we probably never will. So what can we do for those cases of neurodegenerative disease, like sporadic ALS, for example? And this preceded the discovery, really lots of fundamental discoveries surrounding TDP-43. So I'm not going to say that TDP-43 is not a really, really, really good idea. But before that, we thought one good way to go about doing this in a kind of non-specific way, like not knowing anything about like the genetic cause and for sporadic disease, would be to better understand these the programs in neurons that regulate the neurodegenerative processes. And actually, these are really linked to programs that control regenerative processes as well, it turns out. So one example, and the one I'm going to talk about today, is this dual leucine zipper kinase, and the gene name is MAP3K12, really boring name. It's a MAP triple kinase, and I'm going to tell you more about this. So the reason we got into this is because my colleagues at Genentech, this was in Joe Leacock's lab, made a knockout mouse of this DLK. They did that because they had done some screens in neurons to see what genes were really protective when you add something neurotoxic and the neurons die. And they found DLK. So they knocked out DLK in a mouse. And what they found was actually something a little surprising, but it's kind of a cool result. So you may know that some neuron types during development, there's many more of them born than are retained in a mature adult organism, and DLK is really important for that wave of neuronal cell death during development. And that's shown here on the left. You can see on the top row, you've got a wild type animal, and on the bottom row, you have a DLK mutant mouse. And we're looking at HB9, so that's a marker for motor neuron nuclei in the developing mouse spinal cord. And what you can see is that the animal lacking DLK fails to kind of undergo this wave of motor neuron cell death that normally occurs. That's really important for like having a properly mature functional circuit. And these animals actually die perinatally, so we can't study them. But this was really good information, and we asked the simple question, so if DLK is really important to regulate neuronal cell death and in development, which is the normal healthy process, then what about if it gets activated again in the context of disease in a more chronic state? Would deleting DLK or blocking DLK in that case be neuroprotective? And so these are some experiments that I participated in. So what you can see here on the left, this is a wild type spinal cord. We have a choline acetyltransferase immunostain. This is labeling motor neuron cell bodies. And then in the SOD1 G93A mouse model of ALS, you can see those are, this is a mid-stage disease and many of them have been lost. But when we delete DLK, and now this is not the same animal with the germline mutation, this is a different kind of mouse model where we can knock it out later on in life. So normal development can happen. Then you can see that removing DLK was really protective for the neurons. And I'm going to come back to this towards the end of the talk. So just try to remember this one. Okay, so it turns out that one of the best ways to activate the DLK pathway is through axon injury and these traumatic injuries. And so it's a model we use a lot in the lab because it's way less complicated than having like a mouse model of neurodegeneration and try to study DLK in that context. So I'm going to talk to you a little bit about that. And I'm just going to summarize the pathway as we understand it, as we like to think about it now. So you have an axon injury. This somehow will activate this MAP3 kinase, which is it's a very typical kinase in that it will, the active form is a homodimer. So you have two DLK proteins coming together, they phosphorylate each other, and then they phosphorylate downstream targets, including this these MAP2 kinases, and then this MAP kinase, JNK or C-Jun N-terminal kinase. Okay, so this is a classic kind of kinase phosphorylation pathway. But what's really cool about it is that it then can be kind of translated or signal to the nucleus, which we know, for example, in the case of upper and lower neurons, motor neurons, this can be a very long way. The axon can be very, very long, and can transmit this information that, oh, there's been like an event all the way down there and we need to reprogram the nucleus. And so you get this transcriptional response of transcription factors being activated. And this allows the neuron to respond to the injury. Okay, so I alluded to this work before where we showed that DLK also becomes activated in the context of neurodegenerative disease models. And also we see signs of that. It's correlative in postmortem tissue of the pathway being activated in different types of neurodegenerative diseases. So when I talk about axon injury, a lot of times what I'm going to show you is an acute axon injury, but then you can think about how it's relevant because you might, we know that in diseases like ALS, you have different types of stress in the axon that could lead to activation of the pathway. Okay, and then through the transcriptional response, you can have a lot of different things happen. You can have synaptic loss, neuronal cell death, cell survival, axon regeneration. This is just like a jumble of all the things that can happen. It's pretty complicated, but also there's a lot to unpack here. And I'll try to explain what we understand so far about what happens in what context and what doesn't in another. Okay, so I'm going to start off with something that was really, really disappointing to me. This was in 2022. There was the publication that a phase 1 trial for a DLK inhibitor that had been developed at Genentech, GDC0134, had failed. And I had left Genentech a while before, so I had just been very excited for a long time that I contributed to something that's like gone into like clinical testing. It was very exciting. There's a lot of things we don't understand about why it failed. But the trial was halted due to adverse events in some patients, and also, and this was probably the main driver, and was that in the patients treated with this compound, there was an increase in neurofilament light. And this is really kind of like the most commonly used now correlate of neuronal health in ALS. It's a biomarker, right? So this is the citation. You guys can go look it up. But I would argue that there are still many unknowns. And so you might say, okay, well, why continue to investigate this? The clinical trial failed. One clinical trial failed. And I would say there's so many open questions still. We don't really know why it failed. And the thing about the neurofilament light is just really counterintuitive and very intriguing to me. I feel like work needs to be done on that, but I'm not going to talk about that today. So number one, I showed you before that as far as we understand, DLK is really important. If you block DLK, you can protect motor neurons or neuron cell bodies, not just motor neurons. So this could be really important in sporadic ALS. And then targeting DLK to prevent neuron death may actually be very well suited for other contexts of neurodegeneration. We talked about TBI a little bit today. Also in the context of CTE, that could be really relevant for ALS. And then a deeper understanding of the biology around DLK can also reveal other ways to modulate the pathway without necessarily going after DLK itself. And this is really interesting to me. And you'll see right from the get-go, DLK as a target is kind of problematic for the reasons that I will discuss, but I showed you in the diagram. It can do really good things too, like promote regeneration. So why would you want to block that? Okay. So the first, I'm going to tell you 3 little vignettes of recent and current work in the lab, and I'm going to go a little bit fast on this first one because it's all published. So everything I showed you in that DLK pathway diagram is something that the signaling pathway does that's cell autonomous to the neuron. So like it's an effect that happens in the cell that was injured. But the first study that my lab did actually identified a non-cell autonomous role. So something going from the injured neuron towards cells around it. And this work was spearheaded by 2 postbacks at the time in the lab, Josette and Jacob, together with the biologist in my lab, Hannah, who's a whiz at tissue clearing. And I'm going to show you a lot of cool videos that Hannah generated for us. Okay, so the mouse model that we're using here is peripheral nerve injury. So we'll injure one sciatic nerve in a mouse. And it had been known for a long time that if you do that, and so what you're doing is you're injuring the axons of 2 populations of neurons, the sensory neurons of dorsal root ganglia, and also the motor neurons in the ventral spinal cord. And when you do this, about a week later, and you look at microglia in the spinal cord, you see this very, and this has been known before, you see this very pronounced microglial response in the form of these 2 clouds of microglia that are ipsilateral on the same side of the nerve injury. And I'm just going to go back for 1 second. So just an anatomy thing. These neurons here, their axon is out here, but they're pseudo-unipolar, which means they also have a part of their axon that projects into this dorsal horn here, which is where we see one of the clouds. And then these are the motor neurons here, and you guys know those. Okay, so what happens when we do this nerve injury in a DLK knockout? So here now you have a top view of the microglial scar in the spinal cord in a wild type animal. And when we did this in the knockout, lo and behold, it was a huge effect of just we wiped out this neuroinflammatory response. And that to me, and that's as far as I'm going to kind of allude to this story here just for the sake of time. But to me, that was like, oh, this is really interesting in the context of you have a tissue and you have pathology ongoing in the tissue. And if you have DLK signaling, then you're talking to the glia. And we also know that this can also produce, so through DLK, we can activate astrocytes as well. So this is really interesting from a disease pathogenesis perspective, I think. And I'm going to skip over kind of how this, some of the data here, but just really quickly, the way this works is that there's a gene transcriptionally downstream of DLK called CSF1, colony stimulating factor 1. This is actually a cytokine and it's normally not expressed in neurons, but upon injury, the neurons turn it up as part of this DLK response. And so this produces the microgliosis. And we know that because another lab has deleted CSF1 in a few different labs have now kind of like consolidated this work, I would say. So if you delete CSF1 in dorsal root ganglion neurons, you wipe out the dorsal horn microgliosis. we showed that if you delete DLK, you get rid of all the microgliosis. And then since then, we've also now deleted just CSF1 in motor neurons. And here you're seeing just the dorsal cloud in magenta, but you're seeing the injured motor neurons in green, and you see there's no cloud around them. So this is the CSF1 response downstream of DLK. Okay, so we showed that through DLK, you can produce neuroinflammation. And now I want to switch to a newer study where we kind of been digging a little bit into mechanism of how DLK actually acts within the axon. And part of what drove this was because, well, at the time when we started this, we thought, okay, we really have to study DLK in human neurons because there's a clinical trial going on, and everything that's been done has been done in model organisms or cell lines with heterologous expression. And the evidence that I alluded to earlier is from patients is that you will have increased amounts of signaling markers downstream of DLK in patient tissue compared to controls. And also evidence of some of these transcription factors like phosphorylated c-Jun in the nucleus of diseased motor neurons. And the work I'm going to show now has been led by a really talented postdoc in the group, Jorge Gomez Deza, and he's on the job market now. So in case you're looking to recruit someone really good. And what he's shown is a new mechanism for DLK using a neurodegeneration-derived, so cortical neuron system that you've heard about that we learned from our colleague Michael Wards at NINDS. Okay. So what Jorge decided to do in this study was to do a very kind of detailed within a neuron, cut the axon using a laser beam, and then image inside the axon what's going on, and inside the cell body what's going on. So to just see. Most studies of axon degeneration have really focused actually on the other end of the axon. So if you imagine this cut happening here, the distal piece is this piece here. This one undergoes Wallerian degeneration, and we know that this other protein, SARM1, is really important to make that happen. So blocking SARM1 can block the degeneration of the distal axon, but not much has been done on this proximal piece that's still connected to the cell body. So that's what we focused on. Okay. And I'm going to show you some live imaging of mitochondria. And we heard a little bit about mitochondria in the context of ALS earlier because, and the reason Jorge decided to do this was we had been interested in like what proteins associate with DLK. And he had done an experiment to try to label proteins in the vicinity of DLK and saw this really big mitochondrial signature. So hence the interest in mitochondria. And this is the setup in case you're wondering how you do this. So you have your neurons in the dish, underneath this microscope, and then you can actually program the scope to cut the axons at specific places, and then you can image near there at higher resolution what's going on in those cells. Okay, so here, these are two actual videos that are going to play concurrently, and you're going to see something happen very immediately in the green, which is the mitochondria. The asterisk is where he cut. And in the red channel, you see something much slower happening. So did you see the mitochondria kind of just get really tiny? It's going to loop. And on a much slower time scale, the red signal, which is just a red fluorescent protein inside the axon, is kind of disappearing. So we just looped. And the red disappearing is actually reflecting the axon degenerating. I'm not going to demonstrate that here, but we know that. So I'm going to show you a little bit closer up what's happening with these mitochondria. Jorge was very excited. He said, look, these mitochondria are actually undergoing fission. I said, no, they're just getting small. He said, no, they're undergoing fission. And he really had to show this to me. But here's an example from here to here, for example. And here's another example from here to here. And they don't, 100% of them don't undergo fission, but many of them do. And then after this happens, they shrink. What's happening in the cell body? So again, in green are the mitochondria, in red is just this fluorescent protein in the cytoplasm. And the arrowhead is pointing to a cell. So this is a cool video because it's really hard to get this in practice. We have sparse labeling of these neurons, and Jorge found two that were next to each other, and he cut one of their axons and not the other one. So the one with the arrowhead has been cut. So the cell, this is really sped up very quickly, but the cell basically undergoes cell death. it looked like it was exploding. And this is the correlation of if you cut the axon, then the cell dies within about 15 to 20 hours later. So just to hammer home two main points here, we find that with an axon injury, we observe rapid mitochondrial fission, and this precedes a slower axon degeneration that follows. And these both go in a retrograde direction, so from the site of injury towards the cell body. And when Jorge actually decided to look at DLK itself, so DLK now is in green, and I'll play this one more time, he saw something really interesting. So the mitochondria is purple, and DLK is in green, and look at the spot where you saw the mitochondria coming apart. There's some DLK protein right there. So Jorge became interested in kind of investigating this fission more closely. So he deletes DLK from the I3 neurons and figured out a way to kind of quantify this increase in the number of mitochondrial particles after the injury. So that's just normalized to pre and post in a stretch of axon. And when he looked at the DLK knockout neurons, you can see they do end up shrinking, but they don't undergo fission. And this is the graph that can really show that over like a large number of axons and many mitochondria particles quantified. And he finds similar results when he inhibits DLK. And then he further went on to kind of put back into the knockout neurons DLK to just see, can he rescue this? He can rescue this. That's the green curve. But if you put in two different mutant versions of DLK, you fail to rescue it, and that includes a kinase dead version. So one DLK that can't phosphorylate things. Okay, so we knew that kinase function of DLK is important for this. And so one thing that's kind of very well characterized in the mitochondrial biology field is the mechanism of how mitochondria undergo fission. And there's lots of protein machinery that comes together, but a critical piece is this DRP1 protein, dynamin-related peptide 1. It's a GTPase, and it forms these oligomeric, like wrap around both membranes and then brings apart the mitochondrion into two. And DRP1 has to be phosphorylated at the serine 616 residue in order for this fission to happen. Jorge showed that if we knock down DRP1, we see the same phenotype that we see in DLK knockout cells. So this is like the same classic machinery is needed in this case. And so, okay, so we have a kinase and we have a thing that gets phosphorylated. We wanted to know, okay, so is DLK important for phosphorylation of DRP1? And this is just one example of an experiment that Jorge did where he purified DLK, either wild type DLK or a kinase dead DLK, and then he mixed that with a DRP1 protein, and then just looked at the phosphorylation state of DRP. And what you can see here is that if you just have DRP1, there's some baseline amount of phosphorylation. He's producing this from HEC293 cells. And if you add DLK to the mix, you get this big increase in the amount of phospho-DRP1. But if you add the kinase dead version of DLK, then it kind of looks like baseline. So this is telling us that DLK, this is a new function for DLK we really didn't know. The main function had been phosphorylation of the downstream kinase and this kind of canonical pathway that leads to a transcriptional response. And here we have something in the axon very locally, phosphorylation. We don't yet know like definitive proof if it's direct phosphorylation of DRP1 by DLK, but in a sense, it doesn't really matter. It's just like DLK phosphorylation controls downstream DRP1 phosphorylation and causes this local kind of apoptosis, I'm going to call it, in the axon. And there's data I don't have time to show you here, but this is the summary, and this is on bioRxiv, so you can go look. DLK would promote phosphorylation of DRP1. This will cause actual recruitment of backs to the mitochondrial membrane, creating these permeabilization pores that have been well studied in apoptotic biology. And so we have this new mitochondrial DLK signaling interaction, and it's driving retrograde axon degeneration. That kind of reminds me of dying back that you hear described in the clinic or in classic studies. And this at least in our model system, leads to neuronal cell death. Now, and I've said, like, so this is like a local role, an initiation role of like a cell death thing in the axon that's like connected obviously to the cell body, but it's a retrograde mechanism. And I don't have time to show you, but we also have in vivo data showing this can happen, at least in a model of optic nerve crush. And that blocking DRP1 or DLK can protect those neurons. So we're pretty excited about this work. And I encourage you to follow. Hopefully this will get published at some point soon. How much time do I have?
So you have about 7 or 10 more minutes.
Excellent. Okay, time for one more story. So I've talked a lot about DLK having a non-cell autonomous role, and now kind of some nitty-gritty cell biology of how DLK could function locally in the axon. And now I'm going to expand a little bit to refer to kind of, okay, DLK doesn't signal within a vacuum. Of course, there are lots of other proteins and signaling pathways that are activated in the context of injury, and obviously in the context of neurodegenerative disease, and kind of just thinking a little bit about other things that are going on. So I'm going to come back. So I want you to remember what I showed you about that initial study where we knock out DLK in the SOD1 mouse, and we have this really powerful protection of the motor neuron cell bodies. I wanted to see, okay, we got like protection of the motor neurons, but that doesn't mean they're functional. Are they still connected to the muscle? So we started doing these compound muscle action potential recordings longitudinally in the SOD1 animals. And what you see at the top here is wild type animals. They're kind of stable over time between 6 and 19 weeks. And Josette recorded this in the SOD1 model. This is like plain SOD1. So there's a dramatic drop that happens like very early on. So by 9 weeks, where we know that this has been like very well characterized. This is one of the reasons why we did this in SOD1, because like so much is known about what's happening in these animals at specific time points. But at 9 weeks, the SOD1 mice have lost maybe about 50% of their neuromuscular junction connections in lower limb muscles like the gastrocnemius and the tibialis anterior. And so this readout correlates very well. It wouldn't necessarily have to be loss of the connectivity. You could have lots of other reasons why you would have a decline in CMAP, but for this experiment, that's what's happening. So what happens if you knock out DLK in the SOD1 mice? It does not protect CMAP function. So it does not protect how well the muscle can react to a nerve stimulation. Okay, so in these animals, the motor neurons are there, there's more of them there, but they're not connected and they're not functional. And so we looked at the neuromuscular junction, and this is the percent innervated. So like in a wild type animal, you'll have almost 100% of a presynaptic marker in the motor neuron opposing a postsynaptic marker bungarotoxin in the muscle. And then, so this is what you have in SOD1. And when we knock out DLK, it looks the same. So remember I told you about this transcriptional response and how some of these responses could lead to regeneration of an axon. So if you're going to block DLK, you're going to also block some of this good stuff. And one of the ones that I've been always very fascinated by is this activating transcription factor 3, primarily through the work of Clifford Wolff and others. They've shown that in the peripheral nervous system, so now in sensory neurons, ATF3 program is really important for regeneration of sensory neurons. Okay, so we thought, okay, so what if we now put ATF3 back in an artificial way, just overexpress ATF3? So this is what that looks like. So those neuromuscular junctions are well protected. And I have to say, there was a study in, Bob Brown was mentioned earlier. So there's a study from Bob Brown's lab showing, looking at this ATF3 overexpression model in the SOD1 animals, and that had always caught my attention as well. So we decided here to kind of do this combination. So we repeated some of the experiments with the ATF3 overexpression. So you can see this is really good for the neuromuscular junctions. And then if we also cross into that, the DLK knockout, so it's still good, right? So, and maybe this is just driven by the ATF3 overexpression. So what about the CMAP? So this is where we were before. Wild type animals, SOD1. In red, you have SOD1, and we're lacking DLK. So if we just put in just ATF3 overexpression, this is very powerful at improving the CMAP. And then if we combine it with deletion of DLK, that's actually even better. Presumably, this is just my very simple-minded explanation, is you have got more cells there and they are all better connected. So you've got better CMAP. So this is how I like to start thinking about combining or combinatorial therapies, which I think everyone can agree is probably going to be really important for treating ALS. So I want to end on something. So this is an artificial way of expressing ATF3 in these neurons. But what's also been, we've been very interested in and had looked at this for a while, was like just ATF3 expression in an ALS model. Do you see it normally? Do some neurons turn on this regenerative response? And we know, because motor neurons are peripherally projecting, just like sensory neurons, they should have a good ability to regenerate. And just that maybe they're just getting hit so hard in ALS that that can't happen. But we know that from work, for example, Richard Robitaille has shown that at the neuromuscular junction, things are pretty dynamic. And we know from muscle biology and kind of the diagnostic of like muscle fiber grouping that you get kind of more slow fibers kind of trying to compensate probably and innervation of those. So this is pretty interesting. So we decided to visualize the neurons in the SOD1 model that have upregulated ATF3. So what we're doing here, this is a piece of lumbar spinal cord. We have a mid-stage SOD1 animal, and we are expressing green fluorescent protein in neurons that have turned on ATF3. This is using an ATF3 Cree line that we made. And you can see, so just focus, I know the top of the spinal cord can be distracting, and that's interesting too, but this is my last slide. The ventral horn has these very nicely labeled GFP positive neurons that at some point turn on ATF3. And now with this animal, we can kind of look at these. Obviously, this is a fixed sample, so we have to do this cross-sectionally. We can't do it longitudinally, but it's going to be really interesting to understand what is this subpopulation of neurons. And so just really quick, because I'm out of time, this has intersected very nicely with our single cell transcriptomic studies. So a couple years ago, we published an atlas of healthy adult motor neurons in the adult mouse spinal cord. And now we've been doing that over time in the SOD1 model to look for transcriptional changes in the motor neurons of the SOD1 mouse. And so this is an unbiased way of looking for transcriptional changes. And one of the signatures that we see is ATF3. So that made us really happy. So we're working on that, and there will be more to come in future. So just to close, I hope that I've convinced you that this pathway that's really fascinated me for so many years really can regulate neuronal health in growth and development, as well as the death of neurons, and it can play important roles in the mature nervous system and influence disease course. And I've shown you that DLK acts as an injury sensor, and the pathway can regulate axon degeneration, regeneration, neuronal cell death, neuroinflammation. That the pathway is relevant for traumatic injury and across neurodegenerative diseases. And DLK signaling and other stress signals converge. And the reason I probably didn't do a good job of highlighting this, but ATF3 can come on also downstream of other axon signals, not just DLK. So we know there is a convergence there, and there are other pathways also regulating axon degeneration and regeneration. So studying these pathways can reveal future therapeutic avenues. Thank you so much for your attention.
Wow, this was really amazing. And we have multiple questions on the chat. Maybe before people come up to the podium, I'll ask the questions on the chat. Does pathologic DLK activate all three downstream kinases, or junc kinase 3 preferentially phosphorylated? Like, is there a selectivity on which junc kinases are activated, or is it all of them? And the downstream
so
signaling
does DLK
cascade.
phosphorylate all MAP2 kinase,
Yes,
for example?
yes.
No, it's specific. We think it's through MAP2K4 and 7 in neurons,
nice.
and then to junc, to JNK. Yeah.
So the second question, have you localized the kinases downstream of DLK during axonal injury? Have you localized the kinases downstream of DLK
So
during
meaning,
axonal
where
injury?
are they localized?
Yeah, wherever they are
We
localized.
have not looked at them. That's very interesting. I mean, there's so
Yeah.
many questions come out of this immediately. Yeah, don't know.
Okay, maybe question here from the audience.
Excellent talk, and
Thanks.
gives me a lot to think about, particularly with motor neuron subtypes. And in the, I guess in that particular model of SOD1, we know that like the TA muscle tends to be composed of motor neuron subtypes that tend to be more evolved, less sprouting and compensation versus say something like the soleus muscle, which or the gastroc. And in terms of seeing an effect with that, do you think that the motor neuron subtype would interact differently if you had looked at the CMAP and like a plantar flexor
Yeah,
muscle?
really nice questions. We want to do a lot of these things, and I don't have data yet. Those are exactly the lines we're thinking along. Can we look at particular neurons starting from muscle, which we could define as primarily slow or fast twitch? Exactly. Yeah.
Okay.
Thanks for the question.
All right, yeah. Hi, Claire.
Hi.
So you showed that DLK phosphorylation of TRP1 mediates mitochondrial fragmentation and fusion, right? Which contribute to the degeneration. So mitochondria are fragmented, mitochondrial DNA can be released. It has been shown that they are highly inflammatory. They can activate neuroinflammation through the CGAS STING pathway. I wonder, have you looked at it, upregulation of inflammatory cytokines or activation of GGAS STING pathway?
Yeah, so that's really interesting. I'll repeat the question. Basically, that when mitochondria fragment, they can release genetic material, and that can activate some of the neuroinflammatory pathways that have been discussed. But we have not looked at that. And so far, most of what we've done is in this very kind of reductionist 2D culture system of just the neurons. But I think it'll be really interesting. Yeah.
So second question I have is there is a simple DRP1 inhibitor, their DRP1 inhibitor, MD101. So I wonder, have you tried to use that to inhibit the DRP1 to see whether it can rescue or alleviate the detrimental effect caused by DLK or phenocopy or DLK inhibitor?
Yeah. So you're talking about the MDV1 inhibitor?
Yeah.
So we haven't used MDV in the context of these experiments, but yes, I am aware of it.
I'll ask one more question about the second part of your talk, the in vitro model. So in that talk part, I believe with the axotomy, you were seeing some of the neurons were degenerating and having the mitochondrial fission. But I presume some of the axotomized neurons didn't degenerate?
They all do, almost all, yeah.
Oh, I see. And did you have to, I'm actually, because we do sometimes like axotomies and we see regeneration of axons. And so were you trying to target the axotomy really close to the soma or something to get cell death?
Typically in these experiments, we were at least 500 microns away from the cell body. I think when people see something different with an axotomy, it probably has something to do with the culture conditions, the number of days in vitro, what you coat your plate. I mean, it can be so many things, right? So I think the conditions are probably really important, and we're starting to pay more attention to that and try to look at this with other different types of ways of cutting.
Gotcha. Yeah. I was wondering if you had resilient motor neurons in culture, and if there was any.
No, it's not in these cultures.
Gotcha. Thanks.
We have one more question. So this is from Dr. Michelson. MAP4 kinase 4 has been reported to act in parallel.
Has been reported to act in parallel to DLK in axon degeneration and neuronal death. How do you reconcile your data with these observations? So I can
mean,
read it again. MAP4 kinase 4 has been reported to act in parallel to DLK. So they act in parallel in axon degeneration and neuronal death. So how do you reconcile your data with these observations?
Yeah, I mean, I don't think there's anything to reconcile. We haven't really looked at MAP4K. I'm aware of studies from several groups that have shown a role for MAP4K4. Also that it's important in screens for motor neuron degeneration. And I know that I think there are inhibitor programs ongoing to try to target MAP4K4.
Yeah.
From work from Joe Lucock's lab, while it seems like it's not a simple MAP4K is upstream of DLK, despite the name. And so it's not really clear what the relationship is. And in fact, MAP4K might signal through different pathways, but they probably do talk to each other. It'll be very interesting to look at that.
And then when you first opened your talk, you said, you know, there has been one clinical trial, even though it failed. But now you have many more data and you have a better understanding of the biology. So if you were to move forward, what difference would you do in the next clinical trial?
I think we need more research first. And I'm not convinced. I don't have any immediate ideas. I do think that targeting this pathway could be really beneficial in the context of acute trauma. Like if you know you're about to go, I don't know, experience some blast.
Yeah.